Good morning, good afternoon, good day, everyone. My name is Brian Chung. I'm a clinical geneticist. I'm also the president of the Asia Pacific Society of Human Genetics and the chief medical and scientific officer of the Hong Kong Genome Institute. Welcome to the APSHG Hong Kong GI fifth Distinguished Scholar Lecture Series. And as you know, this is a collaboration between the Asia Pacific Society of Human Genetics and also the Hong Kong Genome Institute. And so we would like to bring together renowned scientists, clinicians, researchers to facilitate international knowledge exchange in the field of genomic medicine. And I'm very happy to introduce to you today our fifth speaker, Professor John Christodoulou. Professor Christodoulou is the chair of genomic medicine at the University of Melbourne, the co-leader of the Brain and Mitochondrial Research Group, and director of the Genetics Research Team at the Murdoch Children's Research Institute. His main focus of clinical practice is on the diagnosis and management of children with inborn errors of metabolism, and he has been doing that for over 3 decades. He has an active laboratory-based Rett syndrome research program, as well as in genetic metabolic disorders, especially the mitochondrial disorders. He has a strong research interest in the application of next-generation sequencing technologies for disease gene discovery in rare Mendelian disorders. He is also the executive team member of the Australian Genomics Health Alliance, focusing on bringing NGS diagnostics into mainstream clinical practice in Australia. Professor Christodoulou is a former president of the Human Genetics Society of Australasia, and in 2010, he became a member of the General Division of the Order of Australia, and in 2017 became a fellow of the Australian Academy of Health and Medical Science. Today, the title of his talk is the Australian Genomic Health Alliance Mitochondrial Flagship, a national program delivering mitochondrial diagnosis. John, please.
Thank you so much, Brian, for that very kind introduction. And I'm delighted to be able to give this presentation today. As Brian indicated, I'm from Melbourne. I'm based at the Murdoch Children's Research Institute, which is structurally and functionally very closely associated with the Royal Children's Hospital, which is the major children's hospital in Melbourne, Victoria, and the state of Victoria has around 7 million people. Perhaps one of the things that is a particular standout for the Royal Children's Hospital campus is I think globally we would be the only campus in the world that has our own meerkat exhibit within the hospital. And so this sits in the outpatients departments and helps to distract children before their clinic appointments. So I think what I might do is just reduce this down. That's it. I think this audience really doesn't need an introduction around genomic medicine, or at least in terms of the potential value of genomic medicine in healthcare. And it is a complex process in terms of delivering genomics into healthcare systems, and you can see here the range of activities that need to be considered in order to be able to convince governments to fund genomic sequencing in a comprehensive manner. In Australia, we have a rather complex health system where funding for delivering health is funded at a federal government, and also the states and territories of Australia are responsible for funding certain components. And then, of course, there are private health insurance companies as well. And so depending on what is being delivered in the healthcare system, the primary responsibility falls on one or other of those groups. So, for instance, primary healthcare is the responsibility of the federal government, whereas running hospitals is the responsibility of the states and territories. And then things like subsidization of medications and subsidization of specific testing is funded by the federal government as well. One of the challenges that we've been facing for quite some time is convincing government that genomics is something that really needs to be implemented into the healthcare system. We were fortunate about 10 years ago now, the federal government launched a targeted call for research, which essentially was aiming for a health implementation study focused on genomics. And so the notion was that this project, and we were successful in obtaining that initial round of funding. And so the notion is that this study would demonstrate how patients benefit from having genomic sequencing as part of their diagnostic trajectory. And we, as government always requires, is you need to be able to demonstrate cost effectiveness. So we built in programs to be able to evaluate cost effectiveness. We also wanted to identify where the potential barriers or bottlenecks to delivery of genomics into healthcare would be, and identify potential strategies for being able to overcome them. And then, of course, wanted to build our translational research capacity in the area of genomics. And so there were 4 broad programs. You can see there the National Diagnostic and Research Network, a program focused on how data could be potentially federated and analyzed, the very important program relating to ethical and legal considerations, and then a program focused on education of the workforce. And to gather all of this information, we had a number of streams across all of those 4 programs to gather the data. So we had a number of rare disease flagships and a number of cancer-focused flagships that were used to gather this information. And then as well as that, we also had a reproductive carrier screening stream as well. So this slide shows that this was a truly national project with buy-in from all states and territories, as well as a number of national and international partners. And we're very pleased that Australian Genomics was part of the Global Alliance for Genomics and Health as well. And you can see there the numbers of investigators and partners and committees, and I can't remember how many hundreds of meetings we had over the initial period, but it was certainly a very busy time. A really important aspect of this was the growth of the genomics community or network, social network, over the period of Australian Genomics. And you can see here the initial study at the beginning showing the network of clinical and laboratory geneticists and genetic researchers, and how that grew massively over even a very short period of time. And we haven't done this study subsequently, but I'm sure that that cloud will be even more complex at this point in time. This shows the various rare disease flagships and cancer flagships that were part of this study. And you can see there the numbers of individuals who were recruited into the various studies. All of the genomic testing that was done in a clinical context. So that meant that results were delivered to families and patients. And that's something that we're very proud of. So the data was immediately clinically useful. Some of the outputs from Genomics Australia over the years included, firstly, the development of an online interactive tool for capturing consent in a dynamic manner, so that it meant that families could change their preferences with regards to consent in real time during the course of the project. We developed Panel App Australia, which mirrored to some extent or a large extent the Panel App tool that was developed by Genomics England. This tool is freely available. They're very highly curated gene lists. We think it's popular, as evidenced by the fact that our Panel App Australia gets about 150,000 hits a month in terms of people looking up gene panels. One of the other things we developed was the Sharient platform. And so this brought together clinical laboratories around the country who curated variants, and then they submitted those variants clinically, the details relating to the clinically curated variants, but not the personal details of individuals, into a resource. And if two laboratories submitted the same variant into the resource, but curated them differently, so say one created the variant as likely pathogenic and the other as a variant of uncertain significance, those two labs would receive a ping about that discrepancy, and then they would have an opportunity to discuss their findings. And in almost all cases, consensus was reached around the disease classification of the variant. And this tool also developed the capability to be able to do bulk uploads of data into ClinVar. So all of the clinically curated data has been uploaded into ClinVar progressively. And then the other thing that was a really important piece of work was to develop a white paper for the federal government on how genomic data and the phenotypic data associated with that might be federated around the country to help build practice, not only clinically, but also help build a research resource. And in addition, there were a lot of community-centered activities that took place. So we developed a website that developed and captured information that would be relevant for community members. We developed guidelines to help encourage community involvement in genomic research, so helping with co-design of research projects. And there was also a particular focus on involving our Aboriginal and Torres Strait Islander indigenous populations in this work, because there are a large number of specific sensitivities that needed their engagement to make sure that we were doing this right. And then we also had a program of activity where we started to teach basic genomics in school settings, so to help build the future research and clinical scientists that we're going to be needing in the coming decades. And so Australian Genomics in the first 5 years was very focused on leading research activities, and we were fortunate to get a second tranche of funding. And the second tranche of funding was much more focused on using the infrastructure that had been developed in the first 5 years to help support other research groups in their genomic-focused research activities. And so our federal government had a number of funding opportunities open, focused in genomics, and shown here are a number of research projects that were subsequently funded that drew on resources that have been developed by Australian Genomics to help facilitate their activity. And so there's been a wealth of outputs relating on a number of fronts. And just a couple of those to just mention. Firstly, Mackenzie's mission. So in this study, we looked at expanded couple reproductive carrier testing for around 1,300 genes, and over 9,000 couples who were recruited in this study from around the country. You can see that we went to a lot of effort to make sure that the whole country there was opportunity to recruit from all over the country. And interestingly, we found from this study that about one in 50 couples were identified as a couple of having a high risk of having a child with one of these severe genetic disorders. And most of that risk was not related to the three disorders for which carrier screening is currently available in Australia, namely cystic fibrosis, Fragile X syndrome, and spinal muscular atrophy. And so a wealth of evidence was generated to demonstrate the clinical utility of expanded carrier screening and also the cost effectiveness of doing this. And so we're currently working with the federal government to find a mechanism to hopefully fund this as a national screening program. Another project that was born out of Australian Genomics was our acute care genomics project. So traditionally, genomic testing results take 3 to 4 months, sometimes faster to get back. We were really wanting to push the envelope to see if we could deliver ultra-rapid genomic testing results within a 3-day time frame in the intensive care setting. And so this was a project led by Zornitza Stark, based at our institute, initially a trio exome and then moving to trio genome testing. And we found this to be hugely successful with over half of individuals who were tested in this setting had a genetic diagnosis. And in 3/4 of those cases where a genetic diagnosis was found, it changed the management of those children in the intensive care setting in real time. And that really had a massive cost of benefit, which we predicted, if this was rolled out nationally, would save the federal government $10 million a year just in hospital costs. And so again, we're working with government to develop a national approach to being able to fund that in an equitable manner and sustainable manner. One of our flagships that we focused on was the Cardiovascular Genetic Disorders flagship. So this included individuals with cardiomyopathy, congenital heart disease, and suspected genetic arrhythmia, cardiac arrhythmia disorders. And so over 600 individuals were recruited into this study. And the nice extra component to this was the potential of integrating functional genomics into the diagnostic paradigm was studied as well, including various cell-based models, iPSCs, animal models. And so that was a great success as well. And then, as I mentioned, one of the important things we did was build a genomics community of practice. And so there are just some statistics here. Our Australian Genomics website is live, and there are many tools and resources there that people are free to access and use. And we also had monthly regular meetings, both at our so-called DNA Dialogue seminar series, where we had international speakers presenting. And then we also had a clinical diagnostic and research network monthly meeting where we had Australian speakers presenting on some of their work. And that was really incredibly useful in building a community of practice around the country. So coming back to the flagships, so I mentioned the flagship projects, and what I'd like to do now is move to talking about the mitochondrial disorders flagship. And so I was very privileged to be co-lead that project with a colleague. So just a bit of background around mitochondrial disorders. I would venture to say that mitochondrial disorders are amongst some of the most challenging genetic disorders to diagnose for a number of reasons. One being the onset of the disorders can be at any age. There's broad phenotypic features, as you can see from that cartoon there. And these disorders evolve over time. The traditional approach to diagnosis has been complex, and so there's a broad phenotypic spectrum. And then on top of that, the genetics is complex, with both either nuclear or mitochondrial DNA mutations potentially causing the disorder. And so I guess it's not surprising that the diagnostic odyssey for individuals with mitochondrial disorders has been very challenging. There was a study that showed that many individuals often have to see 8 specialists before a diagnosis is established. Over half were misdiagnosed initially, and nearly a third were misdiagnosed on multiple occasions before the final diagnosis was made. And as I mentioned, the traditional approach to diagnosis is complex. It used to involve, apart from initial screening tests, would often involve moving to invasive testing, muscle and liver biopsies, for instance, which would require, particularly in a child, a general anesthetic, which are not without their own risks. And so even with that approach, there were still problems with regards to genetic diagnosis. And so firstly, the screening tests that were developed or have been used are not so robust. For instance, blood lactate, which is often used, is only increased in about 30% of children with mitochondrial disease. There are several protein biomarkers that can be measured in a quantitative manner in blood that look like they're much better in terms of their sensitivity and specificity for mitochondrial disorders, but they're not routinely available in a clinical diagnostic setting, at least in Australia, across all age groups. And as I mentioned, obtaining a definitive diagnosis requires more invasive procedures, which, particularly in children, requires an anesthetic with their associated risks. And then finally, even if you establish an enzymatic diagnosis through these biopsies, that doesn't necessarily leave anyone the wiser with regards to what the underlying genetic basis of that disorder is. And so that, and again, just to highlight here that, when one thinks about mitochondrial disorders, one thinks about the mitochondrial respiratory chain, but there are lots of other processes involved in generating ATP, and there are genetic disorders, and any one of these can lead to a mitochondrial disorder. And as I've shown this image already, but highlighting the fact that both either nuclear or mitochondrial genomes mutations can cause a mitochondrial disorder. And so there are currently well over 350 different genetic disorders causing mitochondrial disease. So with all of these complexities, that raises the question of whether genomic sequencing, if it was introduced early on in the process, could help achieve a definitive diagnosis more rapidly. And so that led to the genesis of our Mitochondrial Flagship project that was co-led by my colleague of over 30 years, David Thorburn, and I. And so we set up this project aiming to recruit individuals with either pediatric onset or adult onset mitochondrial disease who had not had genomic testing previously. And the other entry criterion was a Nijmegen score of 5 or more. And the Nijmegen score is a score that was developed that captures both clinical information, biochemical testing, histopathology, and any enzymatic testing that was done to score these patients as to whether they might possible, probable, or definite mitochondrial disorder. And then we randomize those to have either whole genome sequencing, singleton whole genome sequencing, or exome sequencing, which, if was negative, then would reflex out to mitochondrial DNA genomic sequencing. And we were using blood DNA as the source of DNA. We recruited about 140 patients into the study, and we used the mitochondrial gene list that's in PanelApp as our starting point. If that was negative, then we would expand out to the Mendeliome, and if need be, we had the capability to apply some multi-omic approaches to help boost diagnosis. And so in terms of our diagnostic rate, we received a diagnostic yield of around 55%. And you can see here the spread of genes that were diagnosed. Amongst those that were known mitochondrial disease genes, 2/3 were nuclear encoded and a third were encoded by the mitochondrial genome. And you can see with a few exceptions, there were no multiple hits of the same gene causing a mitochondrial disorder. An important finding was that in about 30% of cases, the causative disease gene, and they're listed here, was not a mitochondrial disease gene. And it just highlights the marked phenotypic overlap between mitochondrial disorders and many other disorders. In terms of looking at the diagnostic rate of pediatric versus adult onset individuals, we had a much higher yield in the pediatric onset patients, and that's not too surprising for several reasons. Firstly, the genetic testing was done in blood, and we know that for some adult disorders, the genetic, particularly in the mitochondrial genome, the genetic disorder reduces or becomes less obvious in blood DNA. And so you might need to move to other tissues, other sources of DNA to test. And then the second major reason was the fact that we used the Nijmegen criteria for selecting patients into the study, and this table here shows the Nijmegen criteria, and the Nijmegen criteria was developed for pediatric patients. So it's not surprising that it's a much more sensitive and specific test for the mitochondrial onset patients. But what we can say is if someone received a definite clinical diagnosis based on the Nijmegen criteria, then almost always a genetic cause was found, and it was always a known mitochondrial gene. So you can see here, all of these blue dots here represent individuals who had a positive genetic diagnosis, and the diagnosis was in a mitochondrial disease, a known mitochondrial disease gene. Interestingly, the yield from whole genome sequencing was not that much higher than the combined exome sequencing followed by mitochondrial DNA genomic sequencing. We still favor whole genome sequencing for several reasons, including the fact that you're getting both genomes in the one hit with whole genome sequencing. So it makes the whole analysis process much more streamlined. And so this work was published earlier this year in Genetics in Medicine, and the QR code is there available for you if you'd like to download that and read the paper. One of the other things that we did as part of this study was a health economic analysis. And so we were particularly keen to explore whether if genomic sequencing was introduced early on in the diagnostic odyssey, what that impact might be in terms of both cost and diagnostic yield. And so we focused on the pediatric cohort. One was a pediatric so-called pre-genomics cohort that did not have genomic sequencing available to them, and compared them to the pediatric cohort from the Mitochondrial Flagship. And some really sophisticated health economic studies were done, led by a fabulous health economist, Ilias Koronitis, and we showed that introducing genomic sequencing early on in the diagnostic trajectory led to a cost saving per diagnosis of up to $9,000 Australian, and had an increased diagnostic yield of up to 14%. We made an estimate that if this was rolled out at a national level, it would save the federal government close to $1 million a year just in costs associated with making a diagnosis. And so that was published a few years ago. And we're pleased that the data generated from this study and also our cohort study was used as part of an application to make a case to federal government to fund to subsidize the cost of genomic sequencing um, in Australia. And in late 2023, our federal government introduced subsidized funding for trio whole genome sequencing for suspected mitochondrial disease. And so individuals around the country can now have this done at no cost to them, which is, I think, a great step forward for the mitochondrial disease community. So the summary from these studies are we had an overall diagnostic yield of over 50%. Importantly, in about 30% of cases, the gene was not mitochondrial, which highlights the importance of not just looking at the mitochondrial gene panel, but expanding out to other known Mendelian genes. For reasons I alluded to or indicated, the yield was much higher in childhood onset cases than adult cases, and whole genome sequencing had a marginally increased diagnostic yield over the combination of whole exome sequencing and mitochondrial DNA testing. And this is just to acknowledge the huge team of individuals from around the country who are involved in this work. And our funding sources. So not only our national funding organization, but the Mito Foundation, which is a parent-driven foundation focused on mitochondrial disease, helped support this project. We were lucky to have some funding from the US Department of Defense and some donated funding as well to help progress this work. And this is just a photo showing some of the players in the Australian Genomics Health Alliance. And I should have mentioned at the outset that this project was led by Catherine North, who is the director of our institute and has been a phenomenal powerhouse in this whole activity. So I'll switch gears now and talk for a little while about the potential use of proteomics in the helping boost genetic diagnosis. And just to highlight a project that's being led by David, which is a separately funded project called the Mito MDT project, which again has pulled together a national network of clinicians, researchers, and diagnostic groups focused on mitochondrial disease, aimed at applying multi-omics to help boost genetic diagnosis for individuals with mitochondrial disorders. And in particular, I want to focus on our use of proteomics as an orthogonal test. And so it's important to note that different sources of protein can potentially be used to identify in a very highly sensitive and quantitative manner the proteins or protein fragments for thousands of genes. And so if fibroblasts, for instance, over 7,000 genes can be reliably have their proteins identified, which represents about half of the mendeliome. Similarly, for peripheral blood mononuclear cells, around 7,000 genes, half of the mendeliome. And you can see that whilst there's a lot of overlap, there are some genes that will be picked up in fibroblasts, but not in PBMCs, and vice versa. And interestingly, muscle is a less sensitive manner of picking up multiple genes. And so the notion behind proteomics is you take your sample, whatever it might be, and here I've indicated fibroblasts, and you extract the protein, digest it, run it through liquid chromatography, tandem mass spectrometry, and then analyze it. And this gives you an indication of the time frames involved in being able to analyze proteomic data, in this case using fibroblasts. And so just to give you a couple of examples where proteomics has been incredibly helpful, And this has been led, work that's been led by colleagues David Stroud and Daniela Hock, who are based at the Bio21 Institute, which is at the University of Melbourne and is literally a 5-minute walk from our institute. And so this was a child who had a clinical diagnosis of Lee syndrome and had a combination of whole exome sequencing and mitochondrial DNA sequencing. And 2 variants were found. One was a heterozygous variant of uncertain significance in a gene called ATAD3A. And this mutations in this gene can either cause an autosomal recessive or autosomal dominant phenotype. And this child had a mitochondrial DNA variant in the ATP6 gene with 80% heteroplasmy in blood DNA. But there wasn't sufficient data to classify this at anything more than a variant of uncertain significance. So David and Daniela undertook proteomics using fibroblasts from this individual. And what you can see here is a volcano plot. And each of one of these dots is a specific protein that can be precisely identified and quantitated through the proteomics platform that David and Daniela have developed. And so what we show here is if you see proteins in this part of the graph, then that indicates that they're markedly reduced in quantity. And so you can see here that the protein fragments from ATP6 were the most downregulated mitochondrial protein genes. And the other thing that we noted was, and this is often the case when you have a protein as part of a multi-protein complex, you'll often find that other proteins in that complex will be reduced as well. And so there were multiple subunits of ATPase that were also reduced. And so you can characterize that by doing a heat map. And so you can see here that ATPase6 was markedly reduced compared with a number of controls and another patient with a mitochondrial disorder. Whereas ATAD3, which was the other protein we were potentially interested in, showed no abnormalities in its levels quantitatively. And what you can also do is pool the quantitative data for each of the subunits for each of the complexes, and then see whether as a group you see a significant reduction that would give further evidence for the gene being responsible. And so you can see here there was a very statistically significant decrease when all of the ATPase subunits were quantitated together compared to the other complexes and the mitoribosome. And this is a relative complex abundance plot. And then on top of that, for this particular case, we undertook, in collaboration with Luke Formosa at Monash University, non-denaturing Western analysis. And so this is the ATPase complex. You can see here at around 690 kilodaltons. And in this patient, you can see a marked reduction in the holo complex, but accumulation of shorter complexes, which represent a crippled assembly of the ATPase complex. And then the final piece of evidence on top of that was this mother did not carry the mitochondrial DNA mutation in blood, so an apparently de novo variant. So it meant we were able to change the classification to likely pathogenic in this case. So that was very gratifying. And so something that we wanted to explore was whether this technology could be ramped up to be able to deliver an ultra-rapid analysis. And so the limiting factor here, of course, is the source of tissue. And so if you use fibroblasts as the primary source of tissue, it will take, it takes several months from skin biopsy to a fibroblast line being available. And so we wanted to explore whether peripheral blood cells could be an alternative opportunity to be able to undertake proteomic analysis. And so we applied this in this situation here. The processing for PBMCs is only a couple of hours. So technically you could speed up the whole process. And so to test this out, one of the cases we studied was a 3-week-old baby who presented with hypertrophic cardiomyopathy, lactic acidosis, and very rapid deterioration. This baby's parents were consanguineous, and we found a homozygous variant in a complex 1 subunit in an intronic region, so it was predicted to potentially affect splicing. And so we obtained PBMCs from the baby and undertook proteomic analysis, and indeed found that this gene, MBFSH, the gene product, was dramatically reduced, as were a number of other complex 1 subunit complexes. And you can see here from the relative complex abundance plot, there was a dramatic decrease in the quantitative value of all the complex 1 subunits together. And this is a 3D model of complex 1, and just highlights the fact that the subunits in particular that were in close proximity to NBFSH were also reduced. And these results were able to be generated in just over 2 days from receipt of the blood sample. So very demonstrating the ultra-rapid capability of proteomics. And then subsequent work done by collaborators in Sydney, Sandra Cooper and Adam Bonazos, doing RNA sequencing of RNA extracted from fibroblasts from this patient, also demonstrated that this variant did indeed cause a combination of problems, including exon skipping of exon 6 and retention of some of intron 6, and which would be predicted to both cause a non-function of this gene. And this work was just published a few weeks ago, highlighting a number of cases where this technology is very useful. So I guess in terms of talking about mitochondrial disorders, where are we now in the genomic era? There's still we still need clinicians, so don't feel like you're redundant because of genomic sequencing. And we feel that the use of a small number of screening tests could then identify those individuals who would warrant genetic testing, and so you can justify going straight to genetic testing as the next tier of testing. And there will be some instances where the clinical and biochemical picture is so characteristic that you could go straight to a single gene test. And an example of that is if you had a male with a cardiomyopathy, neutropenia, and 3-methylgluconic aciduria, then that is a condition called Barth syndrome, and there's only one gene that causes that, tabazin, or the TAS gene, and so you could make a case for going straight to single gene testing in that instance. But more generally, you could move to trio whole genome sequencing as the next test to do. There will still be some cases where you identify variants of uncertain significance, where functional validation is going to be needed to enable you to obtain a clear diagnosis. So that begs the question of whether whole genome sequencing could be the one test to rule them all, at least for mitochondrial disorders. Switching tacks again, I'd like to talk a little bit about focus on the genetics of mitochondrial disorders and some really exciting work that we're going to be launching in the next 12 months in that space. And so I guess firstly, for nuclear encoded disorders, the current reproductive options in terms of prenatal testing or pre-implantation genetic testing are absolutely perfectly feasible for any nuclear encoded disorder. It becomes tricky, though, for the mitochondrial DNA disorders because of the different mode of inheritance, the fact that heteroplasmy and the bottleneck effect and threshold effects all influence whether someone will develop disease. And so people will be aware that in any given cell, where there's a mutation in that cell, you could have a mix of normal mitochondria and mitochondrial DNA, or mitochondria that have mutant mitochondrial DNA. And there will be a spread across different cells in terms of the mutant load. When it comes to egg cells, this poses the particular challenge. There are about 100,000 mitochondria in a mature oocyte, and there's roughly 1 to 2 copies of the mitochondrial genome per mitochondrion. So there are about 100,000 copies, and depending on the proportion of mitochondria that have mutant mitochondrial DNA, that will influence what might be possible from a prenatal testing or reproductive option perspective. And so if you have the situation where someone has the vast majority of their mitochondria have the normal mitochondrial DNA, then in that instance, prenatal diagnosis or pre-implantation genetic testing may be suitable. But where you have an intermediate or a high level of the mitochondrial DNA mutation, then these technologies become much less reliable in terms of being able to be confident in predicting outcome. And so traditionally, that's meant that the only reproductive option, apart from taking the risk of going through pregnancy, is oocyte donation, egg cell donation. And of course, that's challenging in terms of being able to access enough eggs for to be able to offer this technology. And so this raised the possibility of whether mitochondrial donation could be a potential option in this kind of scenario for this intermediate or high level mutation testing. So what is mitochondrial donation? It's essentially an advanced reproductive technology where one can extract the nucleus from the egg cell of a woman who has the mutant mitochondrial DNA, and so is at high risk of passing that mitochondrial disorder onto her children, and then put the extracted nucleus into a donor egg cell that only has normal mitochondrial DNA and that has had its nucleus extracted. So it's essentially replacing the donor nucleus with the recipient nucleus, and then that embryo then being implanted to allow the pregnancy to be carried. And this technology has been demonstrated to work in macaque monkeys very successfully. And the two main technologies that are available are maternal spindle transfer or pronuclear transfer, with the difference being whether the procedure happens before the egg is fertilized or after the egg is fertilized. So whether it's just the maternal spindle that's transferred or the combined maternal and paternal pronucleus that's transferred. Now, in Australia, the use of this technology has been illegal up until a few years ago because of 2 laws that prohibited this kind of manipulation of human embryos. And so our mitochondrial foundation, the parent-led organization, led the charge at the government level to try and legalize mitochondrial donation. And so this is off the back of activities that happened in the UK. So in 2015, mitochondrial donation was legalized, and the Newcastle upon Tyne group has been running a mitochondrial donation program for a number of years now. And I can disclose, but not any of their results, that 2 papers are scheduled to come out in the next week or two outlining the experience of the Newcastle upon Tyne group using this technology, which I will say has been very favorable. But I can't say anything more because of embargo, unfortunately. But in Australia, the path to legalize mitochondrial donation was a lengthy one, including a number of Senate inquiries and then ultimately conscience votes in our House of Representatives and in our Senate that led to the passing of the laws to legalize mitochondrial donation. And very soon after that, our federal health minister announced a funding opportunity to develop a national program to deliver mitochondrial donation to the Australian public. And I'm very pleased to say that a consortium led by John Carroll at Monash IVF was successful, and David Thorburn and I are co-investigators in this study, and we're very pleased that Mary Herbert, who is a world-renowned mitochondrial embryologist and who was one of the leads in the Newcastle upon Tyne project, has been enticed to Australia, and so she's co-leading this project with us. And essentially, it is a complex beast, as you might imagine. We're currently going through the ethics and governance processes, but once those have all been approved, there'll be the process involved in recruiting individuals with who might be candidates for mitochondrial donation into the study. Then there'll be the mitochondrial donation and IVF procedure that needs to be done. And then hopefully there'll be pregnancies that follow, and then we'll be following up the children following the mitochondrial donation to evaluate them for the clinical outcome. And so there's a whole series, complex series of events that need to take place that is going to be closely monitored, not only by our ethics committee, but also the federal government's Embryo Research Licensing Committee, which will be ensuring that everything that's being done here is done under the most scrupulous and careful activities. So we've recently submitted our clinical ethics application, and hopefully by this time next year, we will be recruiting our first families into this study. And this just lists the investigators around the country who are going to be are involved in this study. So to conclude my presentation, and hopefully, yes, I have left a few minutes for questions. I think genomic testing for mitochondrial disorders, hopefully I've convinced you that it's an absolute game changer in terms of shortening the diagnostic odyssey, will reduce the need for invasive testing, will help restore reproductive confidence, and has health economics benefits. And we're really pleased that in Australia we've now got federal government funding to subsidise genomic sequencing. Multi-omic studies are really critical for boosting the diagnostic yield. And I've been talking about mitochondrial DNA disorders, but we have several other projects that are focused on monogenic disorders, as well as other monogenic disorders, as well as mitochondrial disorders, aiming to gather the evidence to build a case to convince our federal government to fund this as an orthogonal diagnostic test. And then the future really holds fantastic promise, both in terms of advancing reproductive options, but something that I didn't talk about is that there are now internationally over 50 small molecule or gene or genetic therapy clinical trials that are at various stages of operation. And the critical first step in being able to access these precision therapies is to have a precise genetic diagnosis. So genomics is really going to play a huge role in changing the landscape here. And so I'll stop here, and I'm happy to take questions. These two interesting graphics, we asked ChatGPT to draw us some images of what maternal spindle transfer might look like. And this is what ChatGPT came up with, which is somewhat medieval, but we thought it was quite comical. So on that note, I'll stop sharing. And I'd be happy to take any questions if anybody has any.
Thank you, John. excellent lecture. It's very informative. You are telling us all your superb expertise, not only in mitochondrial disease, but in genetic disorders. And you have briefed us in a lot of important details about the latest advances, not only in the technologies that you're using for your patients, but also the evidence-based approach from
Oh, I see
clinical
we have a
trials.
question from Johnson
Yes.
about Mackenzie's mission. So you've been running a similar project, so that's really interesting and a comparable result. Um, and so the question is relating to whether we noticed any differences in the profiles in the East Asian population as opposed to the broader context. I have to say we haven't looked at that, but it would certainly be an interesting question, and it could be very interesting to compare the outcomes of your study against the Australian study. But I can certainly say that the Mackenzie's mission study had individuals from many ethnic groups recruited into the study. So I'm sure there will be at least a proportion of cases in the Mackenzie's mission study were of East Asian background.
Thank you.
And
John,
your question around specific carrier screening gene panels, inevitably, depending on the ethnic group that you're looking at, there will be some potential genes that might be more of a focus than others based on increased prevalence in those populations. So, for instance, I'm aware that in Israel, there is an expanded carrier screening program there, and which has a particular focus on a number of genetic disorders that have a higher prevalence in the Ashkenazi Jewish population, but does include expanded carrier screening as well.
thank you. I think Dr. Low has a question. Dr. Low?
Thank you, Brian. Thank you very much, Professor Christololo, for generously sharing your expertise and latest knowledge in this important topic. I'm the CEO of the Hong Kong Genome Institute, and I have a question relating to your very impressive publications. And as you know, the journal currently requires your raw data set to be made available in public domain. Can you explain how you are handling this?
How we handle disclosing raw data for publication? Yeah, good question. So, um, of course, the data that where a journal requires us to submit raw data, then it's a de-identified data that is submitted. So technically, you can't. Well, I guess ultimately, if you really wanted to, you could potentially identify a precise individual based on their genome. But from a practical perspective, that's never happened. And so where it's required, the raw genome data will be submitted to the journal, but as currently it's held in a federated resource behind a firewall so that you can only access the data with specific permissions. Thanks, Brian. So Brian asked a question about proteomics in mitochondrial disorders, and indeed, what I didn't mention is David Thorburn has been running the main respiratory chain enzymology testing diagnostic lab in the country for over 3 decades, and which has been an incredibly valuable resource. But it turns out that inevitably, depending on the source of tissue that you've got for the enzymology, that you may not get an answer on based on enzymology. And so, for instance, often if fibroblast is the only source that's used for enzymology, you may not get an answer or find an abnormality of the respiratory chain. Whereas proteomics seems to be far more sensitive than respiratory chain enzymology. And David is jokingly talking about the fact that proteomics is going to put him out of business from an enzymology perspective. And in fact, over the last few years, we have indeed seen a dramatic reduction in the number of biopsy samples that are coming to him for enzymology, because people are moving to a genomics first approach and often getting an answer from the genomic result first. And then, if need be, we're in a position where we can offer proteomics as the next second line test in a relatively easily accessible tissue. So having to do major invasive biopsies.
This has been a wonderful lecture. In the interest of time, I'm going to close this fifth lectures on the Distinguished Scholar Series. Stay tuned with our upcoming lectures. Thank you very much, John, for the excellent lecture. Thanks a lot.